Okay, so we're going to be starting in on the cardiovascular section, and we're going to be first talking about blood. And here, if we look at blood, right, anybody remember what type of tissue blood is? From way back in 231. Connective. Wow, you guys remember that. That's awesome. Yeah, it's connective tissue. And remember, the reason it's called connective tissue is that it's got some of its descendants, right, or ancestors, right, were the same as all the rest of the connective tissue. The embryonic ancestor that gave rise to all the connective tissues is the same for blood as is for other connective tissues, which is one of the reasons we consider it connective tissue. All right, and so there's a variety of components that we have with the blood. So you guys are probably familiar with blood cells, and then we have different types of blood cells. We're going to talk about some of the functions. For when we talk about white blood cells and leukocytes, those are really involved with the immune system, and so we're just going to kind of do a very superficial look at those now. We'll take a more in-depth look at them when we get to the immune system in 233. Okay. And so here then we'll talk about something called plasma, and we'll talk about the difference between plasma and serum, and then something called hemostasis, which involves blood clotting factors. All right, so that's kind of the way things are going to go. So we're going to just slowly develop what it means when we have blood, right? So what is in blood? So we have red blood cells, we have white blood cells. What else do we have in blood? Yeah, oxygen. So that's going to be found primarily in what type of cell? Carrying it. Red blood cells, right? Okay. Yeah, absolutely. Platelets, yeah, that's another important thing that's involved with blood clotting, right? Hemostasis, plasma, right? So a lot of nutrients, absolutely. So we're going to talk about nutrients, okay? So not only bringing things to tissues, right, that are important, but what about the other end? So here, what about the stuff that tissues make? How are they going to get rid of the things they make that maybe, if they build up too much, are toxic? Yeah, so they got to be taken out, and one of the major transit systems is blood, right? So what are some of the things that you guys think about that need to be removed from our body that could be in blood? I don't know if our cells make too much of that, right? CO2's one, though. Yeah, so remember metabolism. So you kind of put in oxygen for aerobic metabolism. One of the major byproducts is CO2. That's a major one, right? So, you know, here we're talking about any type of, a lot of metabolic waste and so forth also. So you can't, you know, remember all the good things that blood's bringing to tissues, but also it's helping remove toxic things. CO2 is a major one, but there's other ones out there if you take a look back at metabolism and the things that are made. Endocrine section. What was the major molecule we talked about there? Gets secreted in blood. Starts with an H. Hormones, right? So here, you know, we have also a transit system to move around chemical signals. In the case of the endocrine system, those hormones, right? So that's really important, right? We also find proteins, a lot of proteins in the blood. So it turns out some hormones are also proteins, right? Okay. I know we haven't talked about it, but in the immune system, what do you guys think about when you think about the immune system? What's the major molecule you think about? So white blood cells, absolutely, right? What about a protein structure? So something that binds. What's that? Not a G protein. So circulating in our bodies. That's all right. We haven't talked about these guys. The antibodies, right? So antibodies are proteins. They circulate around in the blood. So there's a lot of substances that circulate around in the blood. Now, what about What do you think? Blood, is it going to be mostly just water, or is it going to have any minerals in it? What do you guys think? So, minerals, yeah. So I think, yeah. So what types of minerals are we going to find in there? Electrolytes, really. Yeah, we're definitely going to get ions, right? So what are some of the major ions we're going to find in there? What's a major extracellular ion? So we have potassium inside cells quite a bit, right? So sodium is the major one, right? So not only do we have proteins, we've got membrane-bound structures like red blood cells, white blood cells, but we also have ions, right? It's really important to remember because when we talk about how blood is going to offload, those things and how it's going to take them in or how it's going to be filtered in the kidneys, you got to remember those, all right? Okay, so we've talked a lot about a lot of these. All right, so you guys may have actually taken a look at this slide before I asked you these questions, which is perfectly fine. Right, so here, we have oxygen, we have CO2, we got hormones, white blood cells, and we talk about antibodies, clotting proteins we haven't talked about much, but we will. All right, regulation, so here we're going to have electrolytes, but we're also going to have certain small molecules that help buffer pH. How many folks remember what buffering or buffers? What does that talk about with respect to pH? Yeah, basically it's preventing pH to change drastically when you add an acid or a base, right? So without buffers, you put an acid and a base and it may move quite a bit. The pH may go up and down, but if you have a buffer in there, it doesn't move nearly as much. So it helps maintain homeostasis, pH homeostasis, right? When we get into talking about respiratory and the renal system, those two systems impact the buffering capacity of blood quite a bit, right? So we have buffers that are going to be in there. Blood volume. So here we're going to talk about that more with the renal system as well, with blood volume. We'll talk about it with cardiovascular because this is a major problem. And then related to blood volume is blood pressure, right? So all of these things are involved with blood. Some of these we'll dig into a little bit more when we get to the appropriate section. Okay, so let's talk about some components of blood. Let's start off with something called formed elements. So why can't I call all of these structures cells? Why do we have to call them formed elements? I think is a good question, right? So here, the definition of formed elements is that it's a membrane-bound structure. That includes cells, because cells have a membrane, right, that binds, that is, going to be a perimeter, which binds all of the things inside of it. But it turns out that platelets, we'll see in the generation of platelets, that they're not cells, but they have a membrane, okay? And so we have to include those guys as part of some of these membrane-bound structures, which is why we can call them formed elements and not cells, right? Because platelets, thrombocytes, okay, aren't really cells. So we got red blood cells, white blood cells, thrombocytes. Some other terminology we have here. So hematocrit. Hematocrit's really a percent of blood that is the formed elements. In general, people use that as a marker for how much of your blood volume is made up of erythrocytes or red blood cells. All right? And we have plasma. This is the blood after we've removed all the formed elements. Okay? So if we remove all of the formed elements, what are some of the things we have left in plasma? So what are some of those other things we said we could find in blood? So we got water, yep. Electrolytes, yep. Hormones, yep. absolutely. Yep. And let's see, what about, say, the immune system? What else would we find circling around? Antibodies, yeah, absolutely. And then also, we haven't talked about these guys quite a bit, but clotting factors or proteins. And they would be left in the plasma, right? So if we remove all of the formed elements, that just means that we're really removing the platelets, the red blood cells, white blood cells, everything that's kind of really big, relatively speaking, compared to, say, the size of a protein. Now, if we go in there and remove all the clotting factors, right, they're involved with clotting of blood, from the plasma, we have serum. All right, so if we remove all the clotting factors from blood, then we have serum, or from plasma, we have serum. Okay? So here, you can kind of think about, all right, we have formed elements, and then we have plasma. Okay, if we remove the formed elements, we're left with the plasma and blood. And then if we remove clotting factors from plasma, we're left with serum. So it turns out formed elements make up somewhere between 37 to 54%, and then plasma makes up the rest. All right? And then here, if you look at plasma composition, most of it's water, but we also have some proteins and other solids. Just because it's a small percentage doesn't mean they're really important, though. Right? Sorry, did you have a question, Tyler? Yeah. Well, we can have proteins that are secreted from cells, so they're no longer bound to the cell or inside the cell. They're pretty much floating free in the fluid. Okay? You following me on that? Okay, so proteins, they can exist inside cells. They can be receptors, like we saw in the endocrine system, membrane-bound receptors, and they can be freely floating. So moving, being part of the plasma there. All right, that's a good question. Everybody with me so far on the components of blood? All right. Okay. So here, components of blood. So we can say that formed elements typically make up about 40% of that volume in males and 42% in females, right? And so here we're talking about generally if that's the percent of formed elements in blood, this is also known as hematocrit. All right? So how do we kind of measure this? Well, what we do is we take blood from an individual and then we centrifuge it. Everybody familiar with this procedure of centrifuging? Okay. So what you do is you take a vial of blood and you spin it around. All right? And so that's going to create a force, and the heavier things are going to be pushed to the bottom of the tube. And then we have the lighter things, the fluid, that stay up on top. And then everything that's in that fluid, such as proteins. All right, everybody with me on that for centrifuging? Okay, so here you spin a tube of, a vial of blood, test tube of blood around really fast. And the larger things like formed elements, okay, are going to pellet compact at the bottom of the tube, and then you're left with a lot of the plasma on the top. Okay. All right, so you do that, and what you end up getting is this part of the tube that's towards the bottom. So remember, we centrifuged this, which means the force was pushing down this way. And the larger structures, right, the formed elements that are bigger, okay, are pelleting at the bottom. And so those are going to be leukocytes and erythrocytes here. Erythrocytes, right, are going to pellet to the bottom here. Then we're going to have this kind of small area that's a bit tan, or it's also referred to as beige, and the original color that it reminded people of was buff. Okay? And in this little band here, right, is where we find our leukocytes and platelets. And then everything above that was the fluid and all the small structures that are known to float in fluid, like proteins. So where would we find hormones, for instance? So here we've got either erythrocytes, this buffy coat, which is named for the color, okay, or up here in the plasma, the fluid. So plasma, right? So these small hormones are pretty small, right? What about antibodies? Where would we find these? So here we find those free-floating antibodies are proteins. So a lot of them, the ones that I'm talking about, are floating around in the blood, the fluid. Or we find that in the plasma, the buffy coat, or the erythrocyte layer. Plasma, right? Okay. What about sodium? Where would I find sodium here, mostly? Plasma. I hope you guys are seeing a trend here. Okay. Right? So a lot of the proteins, these small molecules, right, we're going to find in plasma. Let's talk about maybe you get a drug that's given to a patient and it doesn't bind to anything in the blood. Where would you find that drug? In the plasma, the buffy coat, or the erythrocytes? Plasma, absolutely. Okay, right? So here we're finding, in the bottom we have erythrocytes, then we have a middle layer that's buff-colored, beige or brownish, okay, that has the leukocytes and platelets, and then above that we have the plasma. Right? So here, if we're talking about hematocrit, generally they're measuring what percentage of this whole blood sample is this. So when we talk about hematocrit, we were saying is, you know, for males about 48%, for females about 42%. Right? Okay, so they're saying, all right, the height of this is about 48% of the total height for males, and then 42 or so, you know, for females. Okay, is that making sense? Okay, so. Oh, so if you think about the differences between males and females, so what's one of the tissue differences? If we think about, say, you know, if you look at, say, musculature, yes, exactly. So skeletal muscle. So generally, which sex has a larger amount of skeletal muscle? Males, right? So here, you know, males, they have larger amount of skeletal muscle. What type of metabolism does skeletal muscle do? A lot of aerobic, and then you need a lot of oxygen in order to do that. And so generally that's one of the reasons is you have a little bit more erythrocytes to support that extra oxygen demand. Great question. Yeah. All right. So is that making sense to folks? Right? So you centrifuge blood, right? You can basically compact, or what we say pellet. Okay? You get a pellet, a compact, you know, form of a lot of the formed elements, and then above that we have the fluid component, the plasma. Yeah. Oh, how long before they remix? You know, if you just let that tube sit out, it takes a long time. Yeah. And so remember here with plasma, do we have any clotting factors? Yes. Yeah, absolutely. And so what ends up happening is you probably get this plasma to start to clot before you get mixing that goes on, right? And so there's ways that you can prepare this so that you don't get clotting. All right. So this is a kind of a summary of what I just told you guys about a little bit. In more graphic form, we have plasma, we have our formed elements. Formed elements you can break up into 3 broad structures. Remember, formed elements are just the plasma membrane-bound components. So we have platelets, leukocytes, red blood cells. And so platelets are really tiny, but they're still membrane-bound. They have a phospholipid bilayer. Red blood cells also have a phospholipid bilayer. So do leukocytes. And we'll talk more about those leukocytes when we talk about the immune system in 233. For right now, you know that the leukocytes, there's different types of them. So white blood cells, it's not just one type of white blood cell. There's many different types, and they have different functions. And so here are some of the names, and we'll talk more about those functions when we get into the immune system. And so here, these are actually really, really cool. Okay? So here, some of these we'll talk about when we talk about blood, but we'll talk about a lot more about their function in the immune system. For the plasma, we talked a lot about these already. We have electrolytes, we have nutrients, we have waste, and then we also have some of the clotting factors, so the fibrinogen enzymes are also going to be, a lot of the clotting factors are enzymes. Endocrine system, we have hormones, and then globulins is referring to a lot of the antibodies, and then we have one of the major dominant, most highest percentage of proteins is something called albumin. All right, so albumin is one of the most prevalent protein that we have in our blood. All right, so where do these formed elements come from? Well, they come from the bone, and specifically, which bone marrow? Red bone marrow. All right, so remember we have two types of bone marrow in adults. All right, so red bone marrow, and anybody remember the other one? Yellow, yeah. So yellow is going to be mostly fat tissue, and then we have red bone marrow in adults. That's going to be occupying a lot of the flat bones, and then also heads of the humerus and femur. Okay, where we have primarily our red bone marrow, which means that's primarily where we're getting formation of all of these blood cells in adults. All right, so platelets, another name for those are thrombocytes. So a thrombus is a blood clot, and then leukocytes are white blood cells, and erythrocytes are red blood cells. And so we're going to talk about the formed elements in turn here. So it's starting off with erythrocytes. So erythrocytes are red blood cells, and they have this biconvex shape. And one of the reasons is that during their formation, they actually eject their nucleus. So they don't have a nucleus. High percentage of the dry weight of a red blood cell, not surprisingly, is made up of hemoglobin. We've seen estimates as high as 98% of the dry weight made up of hemoglobin. Right? So we're going to talk about kind of three components of hemoglobin in just a second. But the major function of red blood cells, not surprisingly, is to ferry oxygen to tissues for it to use, and also to ferry, say, carbon dioxide, right, of metabolic waste, from aerobic respiration, back to what structure do we exhale carbon dioxide from? So lungs, right? So here we'll talk about a lot more of this process when we get to the respiratory system. In general, we do not find red blood cells outside of the bloodstream. One of the thoughts is that with this biconvex shape, it allows these red blood cells to kind of squeeze a little bit around tight places. For instance, capillaries, right, where it's really tight and they have to almost go single file to get through this really small blood vessel. Yeah. Yep. Yeah, we'll talk a little bit about sickle cell anemia in just a second. So here, red blood cells, no nucleus. Most abundant protein that they have is hemoglobin. They have very little of other intracellular structures, which means they're really kind of, if you want to think about it, developed, or they've really developed or differentiated to become professional transporters of oxygen. All right, so we've got quite a few red blood cells in our body, so 25 trillion in the body, turnover about 3 million per second. And they're about 8 microns in diameter. They're biconcave discs, like I mentioned. That helps them squeeze through tight places. Also thought to try to increase the surface area so that they can do a more efficient job of taking oxygen up in the lungs and then transferring that to peripheral tissues, and then offloading carbon dioxide from peripheral tissues in the lungs, right? So talked about squeezing through capillaries, and so the general half-life there is about 120 days. And so remember, they don't have the genetic information to do a lot of repair if they do get damaged. And so when I talk about half-life, it means that, you know, if we have, you know, a certain amount of cells, half of them would be lost in about 120 days of red blood cells. Sorry, go ahead, Sabrina. Oh, yes. We'll talk about the disassembly of these damaged cells and the structure where it occurs. Absolutely. Stay tuned. Absolutely. Yeah, it's really important for some of the byproducts that we're going to see. All right. So this is really important, and this is going to lead us into the discussion of how these red blood cells that are old, damaged, old, get disassembled and their products either reused or gotten rid of. For hemoglobin, which is a major molecule that's making up red blood cells, there's three parts to hemoglobin that you need to remember, because it's going to help you with understanding how red blood cells get disassembled, and specifically hemoglobin, which is the major component of red blood cells, get used up or get recycled or removed. One is the globin part is protein. Okay, that's the first part. Right? So hemoglobin, we have a globin part. And then we have something called a heme group. The heme group's known as something called a prosthetic group because it helps this protein carry out its function. All right, so we got globin, then we have the heme group, and then within the heme group we have iron. So those are the three things to remember. Okay? Is that we have a globin, we have the heme group, which has iron in it. Now, each hemoglobin molecule actually has 4 different proteins, not different proteins, 4 protein chains. So we have globins that are the protein chain. So we have a globin here, a globin here, a globin here, and a globin here. Okay? So each one of these globins has a heme group in it to help carry out its function. And each one of these heme groups, it's kind of this reddish circle in the middle, has an iron. Okay? So here the hemoglobin's actually 4 globins, and each one of those globin molecules binds to a heme group, and each side of each one of the heme groups we have an iron molecule, or iron atom. Everybody follow me so far on this? Turns out oxygen binds to this heme group that has iron in it. Okay? So if oxygen binds to, we get 1 oxygen binding to each one of the heme groups. How many oxygens can we bind for 1 hemoglobin? Well, how many heme groups do we have for 1 hemoglobin? 4, right? So again, oxygen bound to this one, to this one, to this one, to this one. So what we say is that this is a subunit, this is a subunit, this is a subunit, and this is a subunit, which means we have 4 subunits that come together to make a functional hemoglobin molecule. All right? So here we have 4 irons, which means that we have 4 heme groups that are going to bind 4 oxygen molecules per hemoglobin. Right? For every hemoglobin subunit, just one of these guys, we get 1 oxygen. Everybody following me on the terminology so far? Okay. All right, so it's important to transport other gases too, so carbon dioxide. So carbon dioxide, for in case those people that are interested, doesn't bind to the heme group specifically. Okay, it's going to bind to another part of the hemoglobin molecule. We'll talk about that when we get into the respiratory section. So what were the 3 parts to remember? What was one of them? Globin's the protein, right? Absolutely. What was the second part to remember? Making up hemoglobin. Heme group, okay, good. And the last part was we have iron in the heme group. I'm telling you, this is going to make your life a lot easier when you talk about this whole disassembling and reusing these guys when you talk about hemoglobin. All right? So keep remembering those are the 3 things that are going to be separated when we break up hemoglobin. All right. Let's talk a little bit about the regulation of formation of red blood cells. So here, this is called erythropoiesis. All right. Okay, so erythropoiesis. Now, surprisingly, this is regulated by a hormone. So you guys just finished up with the endocrine section. So one of the things that you always want to think about is, all right, where is this hormone coming from? From what structure or tissue? And what is the how is that hormone being, what is the stimulus that causes that hormone to be released? And what is the target structure for that hormone, right? So in this case, right, if we have low blood oxygen carrying ability, the hypoxia is the kind of the extreme version, hypoxemia, right? It's kind of the less severe version, and so in the people that study this, they prefer to talk about as hypoxemia, but any type of decrease in oxygen carrying capability, right, to tissues. So here we're talking about if we aren't supplying enough oxygen to tissues that need it, right? That's going to cause a stimulus, right, to generate this molecule called erythropoietin, or from being, it's going to cause this, that is the stimulus that's going to cause erythropoietin to be released from the kidneys. Not talking about the adrenal cortex or adrenal medulla or the adrenal gland. We're talking about the kidneys themselves. They're going to release this molecule into the bloodstream. Where are red blood cells made? Well, all blood cells are made in red bone marrow. So, where do we have receptors for erythropoietin? What's that? Say it again. So bone's the major one, right? Because they're responding to making more red blood cells, right? That's where they're made. And the hormone that's going to be stimulating that is erythropoietin. So enhances more red blood cells to be made, right? And so that's going to restore the oxygen carrying capacity. So we didn't talk about much about the original stimulus, but what are some of the different ways that we can minimize, say, the oxygen delivery? Or if we exceed oxygen, or we have demand exceeded by oxygen delivery, what are some ways that can happen? Some are listed right here, right? So decreased red blood cell count, does that make sense to folks? Right? So what's a situation that could cause that? What's that? Blood loss. Yeah, massive blood loss, right? So you have massive blood loss, which means you're decreasing red blood cell count, which means you're decreasing the ability of oxygen to be ferried from the lungs to the tissues that need it, right? Decreased amount of hemoglobin. So what's a way that that can happen? What's that? Sickle cell will affect the hemoglobin structure, but well, think about the things that you need to make hemoglobin. Iron deficiency, exactly, right? So here, remember, if you don't have the components to build something, you're not going to be able to build as much of it, right? So here, iron deficiency, classic example, right? Decreased availability of oxygen. So what's an example of that? Yeah, elevation gain, right? So if you go up to Mount Everest or K2, right? Oxygen is a lot thinner up there, or, the air's a lot thinner, doesn't have as much oxygen, right? So here, all those things, right, are decreasing the ability of oxygen to be delivered to, tissues that need it. And so that's going to be sensed, and then therefore you get erythropoietin released. So why do you think athletes train at high elevation? So knowing this, what ends up happening? So they go up to high elevation, right? Yeah, go ahead. Right? Okay, so they get increased red blood cell amount, and what's stimulating that increase in red blood cells? What molecule? Erythropoietin, right? So could you do that without going up to higher elevation for those that are bicyclists? Watch the Tour de France, right? So what if you inject yourself with EPO, erythropoietin, right? You get the benefits of stimulating more red blood cells, and therefore you get a benefit of more efficient oxygen delivery, right? Okay, without doing the hard work of training at higher elevations. What are some problems that could happen if we start increasing massively the amount of red blood cells in our blood? What's that? Thickening of blood, right? So if you start adding more and more cells, right, like erythrocytes to blood, you start thickening the blood, which means what structure is having to work harder? The heart, right? And so this is a problem. So this is a major side effect. Right? Thickening of blood. Does it make sense? Right? What we just talked about, how the system works. Where is EPO coming from? Kidney. Absolutely. Right? Major target structure where blood cells are made. And specifically, what type of bone marrow? Red bone marrow, right? All right, cool. So let me just quickly kind of go through sickle cell disease, and then we'll take a break. So sickle cell disease is where there's a mutation in the globin molecule, so that one amino acid is changed. And this is an artist who has sickle cell disease. And he does paintings representing what it feels like when there's a sickle cell crisis. Now, what happens is that in individuals that have only copies of this sickle cell version of hemoglobin, okay, their red blood cells can misshapen into sickle-shaped because the hemoglobin is altered such that the hemoglobin kind of clusters or clumps together inside the cell, deforming the cell. And if you have a sickle cell shape, do you think that that's going to allow easier flow through capillaries, or is that going to be more challenging? More challenging. So it's got kind of sharper edges, and they can aggregate together and cause miniature blood flow problems in capillaries, right? So it turns out individuals that have only the sickle cell, hemoglobin, only one copy of that, and then they have a normal copy of the gene that makes normal hemoglobin, right? Turns out those individuals generally don't have sickle cell disease. So they're said to have the sickle cell trait, okay? And one of the interesting things is that individuals that have that sickle cell gene, a A lot of them are from tropical areas and particularly Africa. And it overlaps strongly with areas that have malaria. So one of the things that happens in the malaria life cycle is that it infects red blood cells, and it can unstress red blood cells. So individuals that have the sickle cell trait, remember they got one copy of normal hemoglobin, they have another copy of the sickle cell hemoglobin gene, okay? They don't necessarily have sickle cell shaped red blood cells unless their red blood cells get stressed, such as if they get infected by this parasite that causes malaria. And then their sickle cell, or their red blood cells become sickle cell shaped. And because they're sickle cell shaped, they are removed from the body, right? And along with being removed from the body, those blood cells are removed from the body, the parasite within them is removed. So it's an advantage to have the sickle cell trait in areas that have malaria because it allows for removal of the parasite. So that's kind of a little bit more information than what you guys need to know, but I think it's really cool to see. Right? And we'll talk a little bit about removal of red blood cells and the breaking down of the components after the break. All right? So why don't we take a 5-minute break, give you a quick intro into talking about hemostasis, which will help you guys with the homework that is going to be used for the in-class discussion on Tuesday. Right? So this is where the payoff is for remembering those 3 components of hemoglobin. So what were they? Protein, globin. Yep. And then what was the second one? Iron. Yeah. Which was part of heme. All right. So this is the payoff. Okay, for remembering that. Okay. So what ends up happening when the red blood cells become a bit old is that they get a bit deformed, and there's kind of narrow and somewhat leaky, we'll say, capillaries that are found in the liver and the spleen. And these formed elements, especially erythrocytes, because they're a little bit deformed when they're older, they get kind of snagged, and then they're removed by certain cells that then digested into those 3 components. Okay? So here, what ends up happening is that we get these red blood cells that are kind of older. They become trapped, in the spleen and the liver. They generally slight deformity in shape, and then it's not quite known exactly how the spleen and the liver determines the age of a red blood cell. All right, but they're trapped in the liver and the spleen there. Now then they start getting broken down. So here, if we have breaking down of those red blood cells in the blood, that's called hemolysis. So here, what we're talking about is we're going to be doing this outside of the blood, and it gets broken down to those 3 components. So globin is made up of protein, and then proteins made up of what subunits? Amino acids, right? So first thing here is that the protein the globin part is going to be broken down into amino acids, which then a variety of cells can use to build other proteins. All right, so that's the first thing, right? Second thing is that we separate the iron from the rest of the heme group, right? So the iron here gets transported back into the bloodstream by this protein called transferrin. It ferries iron back, right? So it can be reused. So iron, minerals, a lot of things are very precious that your body doesn't want to get rid of and wants to recycle them, right? Iron's one of those things, right? So we talked about globin, we talked about iron so far. Last part's the heme group. The heme group gets broken down here primarily in the liver into a variety of structures, biliverdin and then bilirubin. Bilirubin can be bound to that abundant protein found in blood called albumin and transported around the bloodstream. But primarily the bilirubin, if you don't have problems with being able to get rid of it, okay, all right, is going to be excreted into the intestinal tract along with bile. So if you have a problem with your liver to be able to do this, right, then bilirubin is going to accumulate in the blood, and this gives a yellowish appearance. So how many folks have heard of jaundice, right? And jaundice is associated with problems with what organ? Liver. So liver function problems, right? So here, if the liver starts malfunctioning or you're having liver failure, okay, this bilirubin, which is a breakdown of the heme group, accumulates in the blood, and what parts of our body become jaundiced? So the whites of our eyes, right, is one of the places that you can see this. So here it's an indicator of problems with liver. Okay? So here, bilirubin is then excreted in bile into the intestine and then removed via feces. Okay, so there's some intestinal microbiota here that further converts bilirubin into other pigmented structures, which is what gives fecal matter its distinctive color of brown, right? Is this breakdown of the heme group. Remember, how many red blood cells were broken down? Was it per second or per minute? So 3 million. So it's quite a bit. So you're constantly having to replenish a lot of these red blood cells, right? All right, so here, let me just go through a summary of this and I'll get your question. So we got spleen, liver, and bone marrow does a lot of this. Macrophages are a type of immune cell that are really good at breaking down other cells, right? So they start disassembling the hemoglobin, which is the most of what the red blood cell is made of. The globin becomes amino acids. The iron gets reused. The heme group is going to be broken down eventually into this bilirubin, which is excreted into the gastrointestinal tract. So we got amino acids, we have our heme group, and iron, right? So bilirubin, right, can be bound by albumin in the bloodstream, especially if we have liver failure, that amount goes up. Most of the time, if the liver is functioning correctly, that bilirubin is going to be secreted into the gastrointestinal tract. Sorry, go ahead. Mm-hmm. Mm-hmm. There's they may not have this pathway as abundant yet, so they can reverse that, right? Mm-hmm. Yeah, exactly. So they start being able to do this a lot more, right? So that you don't get the accumulation of bilirubin in the bloodstream. Yeah, it's a good question. Yeah. Now, is everybody comfortable then with breakdown of hemoglobin? Yeah. All right. All right, so like I said, I'm not going to talk a lot about leukocytes, right? These are really the only blood cell that has a true nucleus. Remember, red blood cells eject their nucleus. Platelets are just pieces of another cell, which we'll talk about in just a second. And for leukocytes, right, there's going to be 5 major types: neutrophils, eosinophils, basophils, lymphocytes, and monocytes. We'll talk about the function of each one of these when we get to the immune system. All right, so I don't want to have an extensive discussion about that yet. Now, unlike red blood cells, right, these blood cells can leave the bloodstream because they have to go to a site of, tissue damage or infection, and a lot of times that's out of the bloodstream. It's in tissue surrounding blood vessels. So these guys can be able to squeeze through different small structures in between cells to get out of the bloodstream into surrounding tissues where there's tissue damage and or an infection. But as far as the detail we're going to go into for this is that the two major categories of white blood cells, right, are granular versus agranular. So not surprisingly, the granular means that they have granules in them. Agranular means that they don't. The two agranular types are monocytes and lymphocytes. All the rest of them are granular, basophils, eosinophils, and neutrophils. Now, just as a little bit of a preamble to looking at the immune system, a lot of these guys are releasing chemical messengers. Some of these you may have heard of. So how many folks have heard of histamine? So people with allergies like myself, right? Histamine's really bad news, right? Especially seasonal allergies. So here, what cells are releasing these of the ones that we're looking at with leukocytes? Basophils do that. They can release histamine. Right? So these granules that we see are granules of a lot of chemical substances that can be released. Right? So here, phagocytosis is the ability to ingest another molecule. All right? So here, phagocytes are professional eaters. Okay? So macrophages are generated from monocytes, and we'll talk more about that process when we get into the immune system. Now, one mnemonic device to remember all these immune cells is never let monkeys eat bananas. So neutrophils, lymphocytes, monocytes, eosinophils, basophils. Right? So never let monkeys eat bananas. So it turns out for neutrophils, also is a pretty good phagocyte, but these are kind of the nuclear option for getting rid of a bacteria. So they do something that is called oxidative burst. So how many folks have heard of reactive oxygen species? How many folks have heard of antioxidants? All right, so antioxidants are there to help combat reactive oxygen species. So what this cell does is it creates a whole bunch of reactive oxygen species, and in the process it damages a lot of surrounding cells and it kills itself in the process. Right? So neutrophils are really kind of a nuclear option for getting rid of stuff. So that mnemonic device that I told you about, never let monkeys eat bananas, right? It kind of goes down in intensity, starting with neutrophils and then ending up a little bit with basophils. Okay? All right? So granulocytes versus agranulocytes makes sense. Where are these cells made? Where does their manufacturing start? Yep. What type of bone marrow? Red bone marrow, right? All blood cells start off there. Okay? All right. So just as a summary of looking at the difference between red blood cells and white blood cells, right? Red blood cells eject their nucleus. White blood cells, leukocytes, have a nucleus. Red blood cells have hemoglobin. White blood cells do not. Red blood cells transport gases such as oxygen, carbon dioxide. These red blood cells are confined to the blood compartment, and there's one type. White blood cells are leukocytes. They're involved with the immune response. They can leave the bloodstream compartment to the surrounding tissue if need be. There's many different types. Never let monkeys eat bananas. All right? Make sense to folks? All right. So the last formed element we're looking at are platelets. Where do they come from? They come from this huge cell that's found in red bone marrow. It's called a megakaryocyte. What this megakaryocyte does is it has extensions that extend into a bloodstream, and then its extensions pinch off to make platelets. So basically, platelets are just pinched off portions of this megakaryocyte. That's all they are. So you can see why now platelets are definitely not cells, but they're just pinched off versions of this cell, the megakaryocyte. And they have a membrane because they're pinched off from the megakaryocyte. All right? So this is the last formed element we're looking at. Remember, platelets can also be called thrombocytes. So pieces of a pinch from the megakaryocyte. Was a major function of platelets. They're involved with what we call hemostasis. So maintaining the integrity of the blood compartment. Right? So they contain enzymes and also factors involved with signaling for clotting, also recruiting other platelets if there's a site that needs to have clotting happen. They don't last very long, and so here, like I said, the term hemostasis is involved with blood clotting. Okay? So it's a few terms that we've talked about. So we talked about erythropoiesis. That's the formation of what formed element? Red blood cells. Leukopoiesis would be the formation of what? White blood cells. And here we could talk about thrombopoiesis is the formation of what formed element? Yeah, so platelets that are involved in clotting. Absolutely, right? So hemopoiesis is generally all of the blood cells. Okay. Yeah, sorry, go ahead, Tyler. Oh, so where are all of these formed elements made? So where were red blood cells made? Red bone marrow. So these guys are found there. Good question. All right. So let me just give you a bit of brief intro, help you guys with the homework there. Okay. With the whole clotting idea, hemostasis. So if we have some sort of vascular event that compromises the integrity of the blood compartment, first of all, you get pinching in, because remember there's smooth muscle there in the blood vessels, but there's also going to be contraction between cells. Okay? So that's called a spasm. And what this does is it exposes some of the underlying tissue that normally isn't exposed to the blood fluid compartment. Okay? What's going to happen is these platelets, they kind of roll along the surface, the internal wall of blood vessels. And areas that have this vascular spasm, where you have compromised integrity of the blood compartment, they have little molecules that they start sticking out that can bind to receptors on platelets. And so the platelets that are rolling along get stuck to that area. And when they get stuck to that area, the platelets get activated. And they send out signals to recruit more platelets. And then more platelets get to this area, they get activated, and they send out a signal to recruit more platelets. And then those platelets get activated and send out signals to recruit more platelets. What does that sound like to you guys? Positive feedback. That's exactly what it is. Remember, positive feedback is where we have a stimulus, and in result of that stimulus, we add to it. Right? So here we had initial stimulus, which was recruiting of activated platelets, which recruited more platelets, which activated more platelets, which recruited more platelets, which activated more platelets, and so forth. Right? So this happens over and over and over again. Okay? All these activated platelets, not only do they secrete chemicals that activate other platelets and recruit platelets, but they're also stimulating a lot of other factors. Okay? One of those things that is going to happen is, remember, in the blood we have clotting factors. They're also going to be recruited to this area and start a relay, a sequence of events that we call a cascade, where we have one thing that leads to another, that leads to another, that leads to another. Okay, that's a cascade. And so that's what we're going to be talking about a lot on Tuesday, right? So we get damage, we get a spasm, and then we have platelets attached to the damaged tissue, release those chemicals so we get this positive feedback. And the last thing I'm going to show you guys, I'm not going to talk about it, is this. This is the cascade, right? Where we're going to start making a network of these this net of protein, and in it's going to be trapped cells. Primarily, we're going to have formed elements that are platelets trapped in it, sealing off this disruption of the blood compartment. Okay, and this is really part of what the homework has you looking at. All right. Okay, so that's all I'm going to say for now. With the rest of on Tuesday, we'll talk more about this along with the discussion.